So I'm going to talk about viruses mainly today, and this is work done in collaboration with Thierry Morat, who's also at DNS, and Jacobo Marchi and Michael Lessie, who's at Cologne. And then I probably won't get to the part of telling you about the immune system, but if I do, that's work done by Andreas Meyer with Vijay Balasubramanian and also Thierry Morat. All right, so how does this work?
It's with arrows.
Yeah.
No need to work with
No.
the right arrow. Yes.
Okay, good. So probably after this year, you're painfully familiar with viruses and the immune system and the fact that two of them co-evolve together. So the basic idea is that pathogens evolve, they have mutations, but they need hosts to evolve with, so they need immune cells. The immune cells try to kill the virus, so that exerts pressure on viruses, and we have this tug-of-war evolutionary process where we basically is constrained evolution, and we see the collective result of the whole population of immune cells, called the repertoire, against the whole population of viruses in the world. So basically all our immune systems exert pressure on all the viruses that are out there. And the name of the game is, can we predict where the virus will end up? So Michael Lessig and Marta Wuksha, also Richard Neyer and Boris Schreiman and Colin Russell have been playing this game and have been rather successful in it, in the sense that they collect all the data that's out there. This is for the flu at a given point in a year, and they have a model that puts together elements of evolutionary theory with biophysical properties and data about immunogenicity, and make predictions for which strain will be dominant 8 months later, more or less. So they make these predictions in February, because that's when the WHO makes predictions for what viral strain to use. And so they do this really at the whole worldwide level, getting all the data. And what we're interested in asking is, can each one of our own immune systems also make a prediction and have a solution, so basically rearrange the composition of its immune repertoire, which is made out of B and T cells that recognize pathogens, so change the population composition and the density of the different cells in such a way to be prepared for future strains that will infect us later. So maybe I'll tell you about that, or maybe I won't, but first of all, I'd like to tell you about viral evolution. So this is the evolution of influenza A, H3N2, doesn't matter, of one influenza strain over the years. And what you see, it looks very linear, meaning that there's one trunk driving evolution, there's sort of local diversity, but otherwise not much diversity, and clearly there's strong selection.
What's the y-axis? What's the y-axis here?
It's just... The color code is the number of mutations. The y-axis, I think, is just for illustration. We'll get to what axis means in all of this in a second, because that's one of the key things. Okay, alternatively, in the same representation, influenza B, I think it's diversity from this strain, something like that. Alternatively, influenza B looks let me get the right word, qualitatively different, in the sense that it has two branches and there's a clear split that happened in the late 60s, early 70s. So you can see that sometimes splits happen and different modes of evolution are possible. So can we predict which one will happen, and what is the influence of, when they'll happen, and how can we describe it? Okay, so getting to describing all of these things. So what people have often done is they have tried to map, so you get data which is basically a bunch of sequences, and how do you represent it in a way that you can try and make sense of it. So one idea is that of an antigenic map, which is basically a two-dimensional a dimensionality reduction that maps the problem onto 2D, and you plot it in terms of antigenic distance. So what is antigenic distance? Well, you take two viral strains, you put it in the same serum, which is essentially blood of actually ferrets, and you measure the response. So you measure how different is the response of two virus strains to the same blood background. And then you define a distance in the space of response, and then you map this distance in 2D. And so, well, you project it onto a map in 2D, and the first question you can ask is, well, why 2D? And so these are sort of more recent maps for the flu, and ultimately there is no good answer why 2D. Right? So you can ask, so you're getting the idea that I'm talking, well, I'll maybe make this point a little bit more clear, that I'm trying to define, that I'm trying to talk about evolution in a phenotypic space. And an antigenic map is one way to map real data onto some sort of phenotypic space. And another idea that goes back to the 70s and Alan Perelson, or 80s and Alan Perelson, is shape space. So basically this is some sort of formalization or conceptual formalization of the idea that we just saw, that there should be a common abstract space where both immune receptors and viruses evolve. So let's draw it in 2D, because as humans we do well with 2D and we can't really imagine more. And then let's just say that the receptors are spaced, immune receptors, so this is our immune protection, are spaced in 2D. They have cross-reactivity radii, meaning that they have balls around them, that if a virus finds itself in this ball, and this receptor recognizes it, it will still be recognized by the immune system and therefore will die. So basically viruses also get placed in this shape space or antigenic space or phenotypic space. And so antigens and immune cells, so antibodies, live in the same space, and the closer they are in this space, probably in terms of some normal distance metric, the more likely they are to recognize each other. So you see that it is conceptually similar to the antigenic map, that we're basically trying to put things in a dimension that we as humans are happy with, right? And then, of course, this is a picture in 2D. When Perelson proposed this, he says, you know, the first question is, what is the dimensionality of this space? And his answer was, well, maybe 5 or 6. So why 5 or 6? Well, because if I, you know, think about all the biophysical and biochemical properties that a protein has, hydrophobicity, charge, blah, 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 well, 5 or 6, I can make a list of 5 or 6, right? Then other people made lists that went up to 7. But you see that, you know, it's an interesting question. What is this dimensionality of the space? Because I think in this room we understand that things depend on dimension, right? Or do they? So that's one of the things that I'll try to tell you about. Okay, so I said this, okay. And so, okay, so that's sort of the motivation for this. But so what I'll be talking about are mathematical models, or models basically, that try to understand the viral host coevolution and try to get at these questions of how it happens, but worry a little bit about the dimensionality of the space. So the first things we're going to be working in this antigenic space, so I'm going to draw this boxes of phenotypic trait 1, phenotypic trait 2. Basically, you know, this is the space where if they meet, they recognize each other. That's really all you have to know about the space, and that's really all we know about the space. And the big thing is we don't know how to map it to data, right? The antigenic map is the closest thing we have, but ultimately this is something that we would like to do, and I won't talk about today, although I'm trying to do it. All right, so the setup. We have a virus, and we have an immune receptor, immune protection. They live in the same cell. We have these shaded clouds, which are cross-reactivity balls. And so then a virus mutates, and two things can happen. So, of course, when it mutates, it infects other people. Oh, God, this is annoying, sorry. So it can infect a host that already has protection against the mutated strain, and then it'll just clear the virus with existing immunity, so nothing interesting happens. So basically, with probability 1 minus p mut, it doesn't mutate, it'll be the same strain. With probability p mut, it'll mutate, and of course, it can still hit a strain a host that already has immunity, but it can also hit a host that doesn't have immunity. And if this host doesn't have immunity, which we denote like this, basically no protection there, then this host will develop immunity, right? Because that's what happens when you get sick. You develop immunity against whatever infects you. And so we can set up simulations like this, and we can simulate it. And when we do, we actually find 4 evolutionary trends. So we get the trend that we saw for influenza A, which is basically this linear ballistic evolution, where if we look at the sort of lineage structure, we see this we basically see one line, one trend. And the picture that we have here is that basically the viruses are pushed by this population of immune protection. Then, that's where we get this ballistic motion of just being pushed. Alternatively, this motion can be a little bit less ballistic with the virus trying to escape, and diffusive motion. Of course, at the lineage level, it still sort of looks the same. Or we can get splitting, as we saw in influenza B. And this splitting can be transient, meaning that it doesn't live, the split branch doesn't live for that long, or it can be stable, as we see in influenza B. So we're basically able to recover with this model the trends that we see in nature, and we can, sorry, what did this? Okay, so that's what I just said. And we can, analyze the phase diagrams of this, and we can understand, so we can look at different properties. But we can understand that, for example, the average number of lineages you get depends on the mutation rate and the mutation jump. So if you jump far and often, then as a virus, you're more likely to escape the immune systems. And so in this limit, you get this ballistic thing of having one lineage. If you jump far, you can get the many lineages because you're exploring this phenotypic space more often. All right, but that's sort of the introduction really to what I want to talk about. Oh no. Oh no. Oh no. It didn't work, the export. All right, so you have to imagine that behind these things, there's like a green field. And this green field is basically the coverage. So let me start from the beginning. So now what we want to do, so we did the simulations, now we want a coarse-grained model that we can actually make some progress with analytically. And so what we're going to do is we're going to turn to a continuous field description. So we're going to replace our simulations and our agent-based model by fields. So we're going to have a field for viruses, and we're going to call this field N of X, where X is now our phenotypic, many-dimensional phenotypic space. And we're going to introduce an immune memory field H. And so, again, viruses are going to evolve, but now everything is continuous. So the mutations, we're going to assume there's many small mutations that occur often. So high mutation rate, but small jumps. So we're going to approximate this by diffusion. And so we have this equation for the evolution of the viral population, where viruses grow according to some fitness, and then they diffuse according to mutations, and we have demographic noise, which is delta correlated in X and T. we also have an evolution for the evolution of the immune system, where, of course, the immune memory grows proportionally to where the viruses are, right? Because the same idea, you get a virus, after some time, which we assume is fast, you get immunity. And then there's a compensatory term that just basically makes that the total number of hosts is the same. So this is just a normalization term. Okay, so what is this growth term? And now you imagine that there's basically various sort of shades of green, like here, which tells you what is the fitness for the viruses in that given space. And of course, fitness where there is protection is lower when compared to the place where there is no protection. That's what this thing you're not seeing is showing. And what it is described by is immune coverage. So you basically integrate the position of the immune memory with a cross-reactivity kernel, right? With these sort of with these sort of blobs that I was showing before. And then, well, the fitness depends on the susceptibility of a host to a given strain. So it's basically 1 minus the coverage to the power of how many different memories you can have, right? So this is basically how susceptible you are, and then it's modulated by the R0, so the number of injectable hosts. And so this gives you the fitness, which is the growth rate.
Can you explain what is M?
M is the number of memories. So you can have different number of immune memories. So you can think that you can have, when you get infected by a host, you can so for example, you get infected now, but maybe you already got infected by something similar in the past. So that sort of adds to your protection.
But M doesn't evolve.
No, M is going to be a constant. I mean, yeah. It's right. So okay, good. So I'm going to explain what M is in more detail. So first of all, we assume that the host population size is kept constant. So basically the number of possibly infectable hosts. And the number of memories carried by each host is just the integral of the immune coverage, and we're going to assume that that is constant. And that's important. This is where this term comes from, meaning that if I pick up a new memory in a given position, I have to lose another one, right? I can't accumulate memory, so I can't become the super host, well, the super anti-super host that can kill anybody. And so, and this time scale, the integral of this time scale basically describes the immune turnover time. It's the time after which you lose memory again. On average, you lose memory against each host. Conversely, so the number of hosts is kept constant, but the number of viruses is not kept constant. So viruses can go extinct, they can explode, they can do whatever you want. All right, and we measure time in the number of infection cycles. We don't care what happens when there's no infection, but that's basically the setup of the model. Okay, the equation to really keep in mind is this one, which is a simple diffusion equation, right? So it's not too difficult to remember. This equation we're going to quickly integrate out, because I'm going to assume n is roughly constant, and I'll sort of, yeah.
How can you keep m constant since so you have immigration of h? So you have immigration of h proportional to n, right? And n can be, as you just said, n can be very large, right?
Yeah, but it won't be. I'll argue in a second that n is roughly constant.
Okay.
I'm gonna, I'm a physicist. I'll wave my hands and everything will work. All right. So first we simulate this coarse-grained model, and it won't work, will it? No, it won't work. Okay, so if I had my computer, you would see this lovely blue trait appearing because the red dot would move behind it, and then it would leave this blue trail, where the blue trait is basically the creation of the memory of the host. That's the H field, and the red is where the virus is. And if we zoom in, So if we zoom in now into this red dot, that's what you see here, and you see this front of a wave appearing, right? So I'm saying you have to do the animation in your head that this wave passed and appeared, and we have the front of the wave that moves with a certain velocity and has a certain width of the direction of motion and a certain other width in the other direction. And if we, oh my God, if you project this now into the fitness dimension, so you can, this is again in phenotypic space, but now I can project it onto the fitness coordinate, which is here, the y-axis. What I see is I see a fitness wave appearing, a roughly Gaussian-looking fitness wave. And so you can probably see where this is going, because I keep repeating the word wave and wave over again, that we want to link this to fitness wave theory. So I hope you're somewhat familiar with the existence of fitness wave theory. These are things like Michael Desai have done, Igor Ruzin, Herbie Levine, and probably Oskar Halacek, Richard Neyer, right? The idea. So that all that work has been done in 1D. Okay, I actually have all the names written out here. And the thing is now, can we really make the analogy with fitness wave? So first of all, we're more in one dimension, whereas they were in one dimension. But we can deal with that with making an ansatz that we're going to separate out the motion in the main direction of motion, right? So we just follow the direction of the wave, and then all the other dimensions. To make it work, also, we need to assume that roughly there's a constant fitness gradient, so that we can expand this fitness gradient into a constant part and a part that linearly depends on fitness, because we want to put ourselves into this picture where we have these little S histograms in fitness space. But as you see, that shouldn't be too hard. And we're going to assume that the mutations in the direction of motion can either be beneficial or deleterious, but they're isotropically distributed. But in the perpendicular directions, they're neutral. And so in that sense, we end up, well, I'll add a few more caveats, which I'll get to, but I forgot to sort of say, what is the main idea of fitness wave theory? The main idea is that you have this distribution of individuals with different fitness effects, and the ones that are here in the head of the distribution will grow more quickly, whereas the ones here will die more quickly. So this will create a wave that moves with a constant speed. Right? That's the basic idea. And I keep pressing the wrong thing. So with these assumptions, you can actually map it to the fitness wave, where the parameters of our model are here, and map onto, say, the diffusion coefficient in fitness wave and the speed with this scaling in S. And the other remaining problem is that we have a non-constant population size, which produces a constant negative drift. So in normal fitness wave theory, you have a constant population size, and you talk about everything with respect to the mean. We don't have that, but instead we have this S minus SVT term, which essentially adds in the same way as subtracting the mean. This is the fact that the immune system is always exerting pressure, and it's always sort of renormalizing how good you are. So let's talk about this in pictures, and this will sort of help you understand the assumption that I will make. So the idea is now we have this, we have the population of viruses that's moving with a certain velocity. We have the immune systems that follow. So let us assume that we increase the number of viruses, like Amory was saying, that we make them bigger. So what happens next? Well, what happens is that the immune systems jump on them, right? Because you have more viruses, now you have also more immune pressure. So when the immune systems increase, that means that they'll quench the additional viruses, and they'll decrease the number of viruses. And so with these two effects, you end up that there is a fixed point of roughly a constant population size of viruses. Because the host need viruses to move, so you need some viruses, but if you have too many, the immune systems will take over that. So that means that with this characteristic immune turnover time, basically you can roughly assume n is stable. And if you do that and you think back to the diffusion equation, well, n is the integral of small n. If you do that, the diffusion term goes away, you're left with the fitness term. So if you assume that 1 over n dn dt is 0, population size constant, that also corresponds to f0 being constant. So if additionally you make an assumption that the wave is relatively thin, small, compared to vt, so the sort of time it covers, but basically, you take your distribution to be roughly approximated by a delta function. This helps you calculate the coverage to have this form, and then since fitness was 1 minus the coverage, you can write it like that, and then you can Taylor expand to have this linear form where f of 0 has this form. So this, if we now set f0 to 0, this gives us that, which is just algebra. Basically, it's setting this to 0 explicitly. And now I also have tau given from my immune turnover rate. So I replace that, and this gives me a ratio for the virus population size to the speed. Okay, I'm not going to interpret that. I'm just going to leave it for here. I can also calculate the first Taylor expansion term in this approximation. Plug in f equals 0, I get this. And lo and behold, I notice that I have the same terms here and here. So that means that I can rewrite the ratio. I basically take this n and this nh, so I can rewrite the ratio of the virus population to the number of hosts, which is the incidence rate, basically how many hosts gets infected, in terms of the speed of the wave and selection. And this is intuitive, right? The number of hosts that gets infected depends on how fast the wave moves times the selection pressure. But what it gives us is now we have, but it gives us the population size in terms of the velocity, and this is why I spent some time convincing you that I can map the motion to fitness wave theory, because now I can just take the velocity of the wave from the fitness wave literature. Okay, and this is what it scales like. So I'm not expecting you to remember this off the top of your head, because also exactly, what version of fitness wave theory you're familiar, the scaling is slightly different, but it really doesn't matter. At the end of the day, now we have a transcendental equation for n and v that we can solve together and figure out what the population size and the speed of the wave is in terms of the parameters of the model. So we're going to do that. We're going to compare now our theoretical prediction for the number of the viral population size and the speed to numerical predictions. And you see that it works very well. And so basically stop here and say that all these approximations that we made and we sort of, just threw ourselves into, and they're completely uncontrolled, it works. Okay, so we can try and now describe the shape of this wave in more detail. So still staying in this, the direction of the motion of the wave, we can ask what is the shape of the wave in that direction? So what is sigma, what is the width? And for that we can use Fisher's theorem, which connects the derivative of fitness in time to the variance of fitness. And given that our scaling here, goes as sort of linearly in velocity. So in variance it'll go as the square. So basically this is the variance. So we get an s squared variance, and so this should be equal to the velocity, which again scales linearly. So we make the we compare the theoretical prediction with the numerical prediction for sigma calculated this way, and again, it works very well. So sigma is now given in terms of the velocity in the theory. And we can now move on and try and understand something about what happens in the perpendicular direction. Okay, so what happens in the perpendicular direction? We basically have random motion. We have a random, a diffusive walk in the perpendicular direction. Why do we have that? Well, because we're assuming that all mutations are neutral, right? So then the mean squared displacement in that direction is square root of 2d T2, where T2 is the coalescence time between two individuals that we're looking to at how far they got to. And on the other hand, if everything is Gaussian, then the mean squared displacement should be just 2 put sigma put. And so the element we're missing is the mean coalescence time, and that we can again get from fitness wave theory with this funny numerical factor of 166. But it turns out, again, that if we compare the numerical simulations to the predictions, it works very well, up to even this factor of 166 that we recover in our simulations. So it really seems to be a good description for the shape of the wave. Okay, it's 5 to 11. I can just continue a bit. So now, what about the position of the wave? So we have the shape of the wave. What about its position? So let's again try and think about the reduced description. And basically, we now have this vision of the virus being driven by this immune system. So let's just write down an equation for that, where it's driven by the direction and strength is given by the fitness. And of course, fitness we can directly replace by coverage because, we have an equation for that and we can, everything else is basically a constant. So this is our coverage, and so intuitively what we have is we have this picture that we have this ball of immune systems driving our viruses, and if there was no perpendicular motion, if there was no diffusion in the other direction, then it would just go straight, right? The immune system would just push it along and we'd be fine. So that would be this term. This is the inertial term. But additionally, we have genetic drift in the other direction. And so this is this diffusion constant that from time to time there'll be a mutation that takes you out in that direction. And we can define a persistence time asking what is the time that you move along this direction. Wrong button. Okay, so with a little bit of algebra, we can derive this. Or I can wave my hands and just say that, well, what the motion will be is effective angular diffusion in this other direction, where the time scale here combines both the sort of direct effect of the immune system behind you and cross-reactivity. So I set cross-reactivity to 1 in these equations, but that's basically where this equation comes from. And if we basically solve this equation, we get a persistence time for the motion. And if I plug in what we got from setting f0 to 1, I can rewrite this characteristic time, which we can just read off from here, basically, as this. So what goes into this? Well, okay, so first of all, what is this? What does this persistence time depend on? So persistence time is really, physically the persistence time, the time you spend moving in the same direction. And so this is the time for a single virus to escape immunity. And if D is large, so the mutation rate in this perpendicular direction is large, that means that N is also large and so is V, but that will mean that the persistence time is small. So basically, if your population time is large and the speed of your wave is large, then there's many more opportunities for you to go the wrong way, right, with the wrong way is deviating from the course. Same thing if we look at this term, if R is not as large, so the number of individuals you infect, or you don't have a lot of memories, this speeds up the wave, but it'll give you small persistence. So basically, it's like, you know, the faster you move, the more jagged your motion is. So the biological conclusion for that is that for fixed number of hopes, epidemic waves move faster, but also change course more often, making them less predictable. Okay, and then again, we can show that the persistence time is comparable with the numerics. We can now compare this persistence time to another time scale, which basically compares the cross-reactivity radius, so this one, to the speed of the wave. So it asks how much time do you need for the virus population to escape the cross-reactivity radius, so the region where they can potentially get killed. And so this scales as 1 over v, and since v scales as d to the 2/3 plus logarithmic corrections, it'll go like that. But what this time scale is the time scale for the viral population to escape immunity. This is for one virus to escape immunity. This is for anyone in the viral population. So of course, it's a much smaller time scale. And you see the effect, the collective effect of this evolutionary process. Okay, and of course, this is driven by fast mutations because it's driven by the motion in the mutations in the direction of motion, and this is driven by neutral perpendicular mutations, and that's why it's slower. But both of these time scales are much larger than the coalescence time scale, which is the sort of ultimate underlying evolutionary process. And that's, you know, things coalesce very quickly, but then you renormalize by the effects of the immune system, and this gives you the larger immune systems. Okay, so last points are about predictability and splitting. So can we predict where the virus is going in the sense of not just direction, which was what T-persist was, but really can I predict where the virus will be in antigenic space in the future? Okay, so how do we do that? Well, we ask, we compare the variance in the position to the cross-reactivity range. And so we now understand we have angular diffusion in this other dimension. So we can write down, well, we can derive this, but I can sort of wave my hands and say I just write down the scaling for the variance of the position with angular diffusion. Solve this, and you see that the prediction timescale now depends on dimension. So that means that in higher dimensions, you have more possibilities for deviating from the course, so things are less predictable. And interestingly, in small dimensions, the timescale for predicting things correctly, the position of the virus, is between the persistence timescale and the escape timescale. It's actually its exact geometric mean. However, in large directions, the prediction timescale is really very small because, again, very quickly do you decorrelate and you can end up going in a completely different dimension.
Escape is population escape.
Sorry?
T-escape is population escape.
Yeah, T-escape is population escape. So the last thing is wave splitting. So we can ask after what time will two viruses end up far enough away from each other? And again, far enough away from each other we'll say is larger than the cross-reactivity range. So this requires a little bit more work, and you do a mean first passage time calculation, and you get an exponential dependence on dimension now. So again, you can see that it works compared to numerical simulation, but the splitting rate grows with dimension. So that means that departure from canalized evolution is easy in higher dimensions. Again, we talked about this at the level of numerics. We're not surprised, but it's interesting to see how this compares to all the other timescales. And the answer is that splitting, since it's an exponential process, as opposed to being power law, having a sort of power dependence in all the other cases, splitting is much less likely than deflections. So, which agrees with what we see in the data, but also puts a precise scaling with dimensions on all of this. So, to summarize, since none of this will work, well, we end up with these different characteristics. We end up with a link between the population size and the wave velocity. There's a picture in terms of the wave. And we get timescales for persistence splitting, the population escape, and the sort of persistence scale. And we can sort of put them in order, which I discussed. And we have these scales that really depend on the effect of the immune system and a timescale that really doesn't. Okay, and then we can try, and that's what we're doing right now, try and go to data and see whether we can extract some of this from sort of population-wide and sequence data. So I have part 2, but I'm going to stop here because I think we're over time.